Okay, let's just start with this. Imagine a single faulty gene, just one tiny dominant mutation,
Right.
but it acts like this cellular time bomb. Simultaneously attacks your skeletal muscles. It starts to dissolve your bones, causes catastrophic neurodegeneration, and then on top of all that, it leads directly to heart failure.
It's an absolutely brutal web of destruction. And that's exactly what defines valosin-containing protein, or VCP, associated multisystem proteinopathy. Clinically, we call it MSP1.
And today we are diving into a really comprehensive new review article that's trying to build a clinical framework around this.
Yeah, because for far too long, the cardiac component of this disease has been critically underrecognized.
Even though it's often what leads to mortality.
Exactly.
So the complexity, it really starts with the protein itself, VCP.
Right. It's also known as the endoplasmic reticulum ATPase P97. It's a rare autosomal dominant inherited disease.
But while the disease is rare, VCP's job is it's everywhere, right? It's expressed in every single cell in the human body.
Yeah. Ubiquitously. And if we had to give VCP a title, it would be something like the ultimate cellular housekeeper.
The universal recycling manager, maybe.
That's a great way to put it. It's a critical regulator of cellular homeostasis. I mean, think about the cell's entire waste management system.
The plumbing, the garbage disposal.
All of it. The ubiquitin proteasome system, ER-associated degradation, autophagy, mitochondrial quality control. VCP is running the whole show.
And this umbrella term, MSP1, that covers the classic symptoms we hear about.
That's right. MSP1 is the catch-all for that cluster, which you might see referred to by the acronym IBM-PFD.
Which stands for inclusion body myopathy, Paget's disease of bone, frontotemporal dementia,
and/or amyotrophic lateral sclerosis, ALS. The source material is clear. VCP mutations are the established cause for that whole spectrum.
So if this VCP is the cell's, universal extraction and recycling unit,
Yeah.
our mission today is to unpack what happens when that unit fails specifically in the heart. Because the review strongly suggests that even if a patient is diagnosed with the muscle or the neuro issues, the cardiac component, which is so often silent, is the thing that can kill them.
And that silent progression is exactly why proactive screening is so vital.
Okay, so let's get into VCP's normal function. You called it the recycling manager. How does it actually move things around?
The mechanism is pretty amazing, actually. VCP is an ATPase.
Meaning it uses energy from ATP.
Right, it uses the energy from breaking down ATP to basically force and release proteins. Think of it like a microscopic molecular machine, a tugboat.
Pulling stuck garbage out of a massive pipe.
That's a perfect analogy. It latches onto misfolded or unwanted proteins and literally pulls them out of big cellular structures like ribosomes or chromatins.
And once they're pulled out,
It makes sure they're ushered over to the disposal units, the proteasome or autophagic pathways, to be degraded. The whole point is preventing toxic protein aggregation.
so the disease is basically a failure to clear out that debris. Impaired proteostasis.
That is the core of it. When the VCP variants show up in cardiomyocytes, the heart cells, that's what happens. The clearance system breaks down.
And you get this cascade of failure.
Yes. The mutation impairs protein degradation, you get chronic misfolded protein stress, and critically, it messes with mitochondrial function.
The cell's power plants.
Exactly. And all those failures compound, leading to electrical instability and eventually what we call maladaptive cardiac remodeling.
What's really fascinating here is how VCP cardiomyopathy is so different from other inherited cardiac diseases we talk about. It's not just a structural problem.
And that distinction is key. When you look at diseases like desmonopathies or CRY-AB mutations, those are often about structural damage. They disrupt filaments, they cause visible protein aggregates.
Right.
VCP is a different beast. It targets the process.
So back to your analogy, it's not breaking the beams of the house, it's stopping the trash collection service.
Perfect. It impairs proteostasis and quality control. So you get dysfunction, diastolic, systolic conduction issues without one single defining histological lesion. It's the cell's inability to cope with stress.
And the real irony is that when VCP is working properly, it's not just passively cleaning, it's actively protecting the heart.
Absolutely. The animal models show this so dramatically. Researchers engineered mice to overexpress normal VCP just in the heart.
And what happened when they stressed those hearts?
They subjected them to ischemia reperfusion injury, which simulates a heart attack, and the protective effect was astounding.
How significant are we talking?
A 50% reduction in the size of the myocardial infarcts.
50%.
It's massive. And it comes from VCP promoting cardiac survival, partly by upregulating nitric oxide and improving mitochondrial function. It's a true cardioprotector.
The second that mutation gets introduced, that protection is just gone.
It's wiped out, and it's replaced by an inability to adapt. That maladaptation seems to be the defining feature of a VCP disease in the heart.
So what does that look like in the lab?
Well, look at the studies where they induce pressure overload on the heart by narrowing the aorta. It's a procedure called transverse aortic constriction.
And in a normal heart, that pressure makes the heart muscle thicken to compensate, right? It gets stronger.
Exactly. A healthy adaptive change called left ventricular hypertrophy, or LVH. But in the mice with the VCP mutation, they didn't develop that preserved hypertrophy.
What happened instead?
They quickly developed dilated cardiomyopathy, DCM, and their heart function just plummeted. The mutation fundamentally cripples the heart's ability to undergo positive remodeling.
And there was one specific transgenic model, the K524A mouse, that made this even clearer wasn't there.
It did. That model developed progressive cardiomyopathy with systolic dysfunction and an enlarged heart, atrial dilation, all within about 9 months.
And the key finding?
The key finding was that this happened without significant LVH or major structural remodeling. It just confirmed that the faulty VCP limits the heart's intrinsic ability to adapt to stress, long before you even see major scarring or dilation.
Okay, let's bring this into the clinical reality. The scope of this problem is, it's severe. Patients with VCP, MSP face progressive motor dysfunction, quadriparesis, respiratory failure.
And when you couple that with cardiac failure, it often results in death tragically early, usually between ages 40 and 60.
The prevalence is hard to pin down, I imagine.
Very hard. It's a rare disease. But the cardiac risk is substantial for those who have it. One family study showed 27% of affected individuals developed cardiac issues.
And another study looking at echocardiograms.
A smaller review, but it found a 43.5% prevalence of diastolic dysfunction in symptomatic patients. That's nearly half.
And when they image these hearts, they're seeing both types of major structural failure.
Yes, both dilated cardiomyopathy, DCM, where the ventricle gets big and weak, and hypertrophic cardiomyopathy, HTM, where the walls thicken abnormally.
That variability plus the conduction issues like atrial fibrillation just speaks to the chaos this protein failure causes.
It really does.
For a patient who's already struggling with, profound muscle weakness, what are the specific cardiac symptoms a clinician should be looking for?
Well, initially they might be completely asymptomatic, and the big problem is that the motor decline often overshadows the early subtle cardiac symptoms.
They just assume they're out of breath because of the myopathy.
Exactly. But as the cardiomyopathy progresses, they will report classic heart failure signs. Progressive dyspnea, shortness of breath, exercise intolerance, and signs of volume overload.
Like edema.
Lower extremity edema, elevated jugular venous pressure, things like that.
And that symptom overlap leads right into the huge problem of misdiagnosis.
It is a profound challenge. VCP disease is so often mistaken for more common neuromuscular disorders.
Like what?
We see misdiagnoses like facioscapulohumeral muscular dystrophy, myotonic dystrophy, or even ALS. And this delay, which can last for years, is just catastrophic because it prevents timely intervention, especially for the heart.
Okay, so given that challenge, we need standardization. But I understand there are currently no VCP-specific evidence-based guidelines for cardiac care.
That is correct.
Yeah.
I mean, VCP patients are, by necessity, excluded from large clinical trials. So the current approach relies entirely on expert consensus and basically extrapolating from other types of heart failure.
So once a VCP diagnosis is finally made, what does that consensus recommend for a baseline cardiac screening?
You need a multidisciplinary team cardiologist, neurologist, geneticist. A baseline evaluation of all organ systems is crucial, with annual screening suggested after that.
And for imaging?
Well, an echocardiogram, an echo, is standard, but it has known limitations in these patients. Skeletal abnormalities like kyphosis or scoliosis can, block the acoustic windows.
Right, it makes it hard to get a clear picture, which is why the consensus points to a more advanced technique.
Cardiac magnetic resonance imaging, or CMR, is often considered the gold standard here. It gives you accurate dimensions, function, and tissue characterization.
So the expert guidance is to start with a CMR and an ECG.
Yes, and if that's all normal, you'd suggest follow-up monitoring maybe every 2 years.
But what if they find something, some early damage or scarring?
If abnormalities are detected, like ventricular dysfunction or the presence of late gadolinium enhancement,
Which is the imaging hallmark of scarring.
if you see that, then you need much closer follow-up. 6 monthly clinical check-ins, maybe an annual CMR. But we have to stress, this is all interim expert-driven guidance. It's not based on VCP-specific trial data.
And when it comes to treatment, it's the same story. Therapy is essentially borrowed from the latest guidelines for non-ischemic heart failure with reduced ejection fraction.
That's right, the 2022 AHA CCFSSA guideline. The treatment is built around what we call the four foundational pillars of HF-RDS therapy.
Let's walk through those. What's pillar number one?
The preferred first step is usually an angiotensin receptor neprilysin inhibitor, or ARNI. It's generally favored over older ACE inhibitor, an evidence-based bull blocker. Helps slow the heart rate, reduces stress on the muscle. A mineralocorticoid receptor antagonist, an MRA, a specific type of diuretic that has powerful mortality benefits.
And the fourth, which is a relatively newer addition but a crucial one.
That's a sodium glucose cotransporter 2 inhibitor, or SGLT2 inhibitor.
And that's used regardless of whether the patient has diabetes.
Correct. These drugs have shown really substantial cardioprotective effects in non-diabetic HFrEF patients, but the challenge remains. We are treating the consequence,
Pump failure,
not the underlying VCP-driven protein failure.
which just highlights the urgent need for targeted therapies. This management saves lives, but it does nothing to stop the relentless neurological and muscular decay.
It doesn't, and future research needs to go way beyond just symptom management. We need to identify biomarkers for early risk assessment.
And on the molecular side,
We need to fully elucidate the specific pathways, like mitochondrial quality control and how VCP impacts mTORC1 signaling, which is a master regulator of cell growth.
and a huge gap the review pointed out was the lack of genotype-phenotype studies for the cardiac side.
It's a massive blind spot. We have data for the neuro and bone pathology, but no one has really looked at whether certain VCP variants have a higher prevalence or severity of cardiomyopathy.
We need that data to tailor strategies.
Absolutely. Using that high-resolution CMR imaging we talked about is essential for that.
Now, despite the lack of current drugs, we have seen some promising preclinical work aimed at fixing the gene itself, haven't we?
There's been some encouraging work using a mechanism called exon 5 skipping in mouse models.
To basically excise the pathogenic variant.
Exactly. And it was very successful. It improved muscle strength, reduced brain pathology, improved bone density.
But the perennial VCP problem.
It pops up again. It's critical to note that the cardiac parameters were not assessed in that study.
So we still don't know if that genetic fix actually helps the heart.
You don't.
And finally, the genetics here suggest VCP isn't acting alone.
That's a crucial point of nuance. The fact that some VCP mutation carriers never develop cardiomyopathy suggests there are other factors at play.
Genetic, epigenetic, environmental modifiers.
Yes. But what's really fascinating is a correlation they've found. VCP variants linked to an earlier disease onset tend to exhibit higher intrinsic AT-pace activity.
So the problem isn't just that the housekeeper is broken. In the most aggressive forms, the housekeeper might actually be hyperactive, burning energy too fast and causing chaos.
Exactly. It might be overcompensating, which leads to a more rapid, more destructive disease course. And that could be a potential therapeutic target.
So to summarize our deep dive, VCP cardiomyopathy is this distinct clinical entity. It results from these fundamental cellular failures, impaired proteostasis, mitochondrial dysfunction that show up as the heart's inability to adapt to stress.
And because of its rarity and multisystem nature, proactive, specialized screening with tools like CMR is just crucial, even though our treatments are still borrowed from standard heart failure guidelines.
Which brings us to a final provocative thought.
And this is something for you to really mull over as you process all that. Given that the VCP mutation prevents the heart from adapting to normal stress, and we know that the more aggressive variants have that higher AT-pace activity,
Right.
how might the development of specific biomarkers, ones that could signal that elevated AT-pace activity, lead to truly prophylactic interventions years before any symptoms even appear? Could we fundamentally change the prognosis for these patients from, a death sentence to a manageable condition?
Keep asking questions, keep exploring, and definitely keep diving deep with us.